IBC-Ab002 is a first-in-class treatment, potential treatment for Alzheimer's disease. It is a monoclonal antibody that is directed against the PD-L1 immune checkpoint. We are all familiar with the use of immune checkpoint inhibitors in the treatment of cancer. This is because cancers, being very clever, have adapted a mechanism that normally our bodies have to control the immune system, to balance our ability to fight off invaders, and balance that with the tendency to overreact and actually generate autoimmune diseases. But cancers have learned how to evade detection in the body using these immune checkpoints. But that's not why we have immune checkpoints. They're there to help our immune systems control and regulate their activity. As we age, our immune system becomes what we call senescent or exhausted. This is why older people are susceptible to all kinds of diseases. So the fundamental question we have is, why can't the body fight off the amyloid and tau pathology of Alzheimer's disease? And the answer may be that because the brain relies on the immune system all throughout life to help it maintain its normal state of health and repair, that as the immune system weakens, the brain and the body lose their ability to fight off the challenge of Alzheimer's disease. So if we could just turn up the activity of the immune system a little bit, maybe we could help that process along. Now, the brain does have its own immune cells. These are called microglia, but it turns out that as the disease goes on, microglia also get exhausted. And because they're working so hard to try to take care of the amyloid and the tau in the brain, they get overactive and they actually start causing damage. So this process of exhaustion in the immune system turns out is in part an active process. And these immune checkpoints, you can think of them as a lock and key, where the lock is on the immune cell and the key is on the antigen presenting or other cell types in the body. When these two engage, the immune system can't react. And so if we block this with an antibody, the immune system can become more active. And what happens then is that this, and maybe I will share at this point, the immune system becomes, and I'm going to share from the poster that we presented here at CTAD. When the immune system, when the T cells are activated by blocking, if you can see me pointing here to the PD-1, PD-L1 checkpoint, they become active and they start releasing gamma interferon. And we know from prior work of others, for example, Rudy Tanzi and his group published a very interesting paper in last winter, showing that in people with early Alzheimer's disease, the levels of interferon gamma in the body are low. Why is this important? Well, it turns out that the brain has a number of very specialized regions that we call the brain border regions, and these include the choroid plexus, specialized areas of the meninges, etc. And these areas surrounding the brain require gamma interferon to allow immune cells from the bloodstream to come in and perform immune surveillance in the brain. So that when we block the PD-L1 checkpoint, particularly monocytes from the blood transit through the CSF and the brain border regions and enter the brain. And once they're in the brain, they acquire several very special properties, including the ability to, because they have what we call scavenger receptors, to gobble up amyloid and tau oligomers in ways that microglia can't. So they start helping the microglia. And they also, it turns out, reduce the level of inflammation in the brain at the same time. They help the microglia calm down. So you have them helping the microglia, doing things that the microglia can no longer do, but also help normalize the function of the microglia. And we also bring in what's called regulatory T cells. These are cells that also have anti-inflammatory properties. So when this method of treatment was tested in animal models, it's been tested now in 7 different animal models. I hope the people on the screen can see this. Several in IBC's lab, or the laboratory of our scientific founder, Professor Michal Schwartz at the Weizmann Institute, and 3 independent laboratories. We find that in amyloid transgenic animals, we reduce the amyloid pathology, and in tau transgenic animals, we reduce the tau pathology, which is very interesting because we have one treatment now that is reducing amyloid and tau. And this happens even in animals that lack TREM2, so their microglia are not active. And when we do this, we rescue neurons, we reduce inflammation in every model where it's been tested, and we have improvements in cognitive performance. In mice, this is usually testing animals in a maze. So we have one treatment that simultaneously reduces inflammation, reduces amyloid, reduces tau independently, and improves behavioral manifestation in Alzheimer's disease models. And what's more interesting, and what's not shown in this poster, we showed it in previous posters, is that this effect is very long-lived. And you can dose an animal with an anti-PD-L1 antibody similar to IBC-002 and find that cognition is improved and signs of pathology are improved as long as 1 month or even 6 weeks later, so that in animal experiments, we can dose this antibody once every 6 weeks. And what's more, because we only need to dose the antibody very infrequently, because what's doing the work for us are these monocytes that become macrophages in the brain, we can work with an antibody that has a very short half-life. And when we do that, we believe that we can reduce the risk of autoimmune side effects that have been seen when cancer patients are treated with immune checkpoint antibodies. The other very, very nice thing about this mechanism of action is that unlike amyloid antibodies and tau antibodies, the target engagement, that is the binding of the antibody to the molecule, to the molecular target, is happening in the bloodstream. So we can measure in animals and in people, we can measure the levels of antibody in the blood. We can measure the binding to T cells, which actually maximizes several days after the dose. We can measure the saturation of the target. We can measure the activation of the T cells, and we can measure the activated monocytes that are destined to go into the brain. So unlike lecanemab and all the other amyloid antibodies, we can measure this very easily, which will help us set our dose for the clinical trials. And then in laboratory experiments, Professor Schwartz and her colleagues have shown and published in journals like Nature and Nature Communications and the like, that we see a decrease in the bad cytokines IL-1, IL-6. We see an increase in anti-inflammatory cytokines in the brain. We see decreases in levels of tau in the brain and in CSF. We see preservation of synapses that I don't know you can see too well here, and of course, improvement in behavior. And in our clinical trial, we can measure changes in cytokines and amyloid and tau in the blood and in the spinal fluid of our patients. We can measure the function of synapses using quantitative EEGs. And of course, even though the numbers of patients will be small, we can use cognitive tests, and we'll be using cognitive tests that are very, very sensitive to functional changes in the early stages of Alzheimer's disease. And furthermore, the fact that we don't need to dose very frequently will enable us to dose our patients only once every 12 weeks in this clinical trial. This is very convenient for patients. It should minimize adverse effects and should enable us also to be very conveniently combined, for example, in the future with anti-amyloid treatments, which is important because lecanemab, as beautiful as the data was, only produced about a 25 to 30% improvement in patients, which means that we have a long way to go before we can completely arrest the ravages of Alzheimer's disease. And the hope is that treatments like IBC-Ab002 can either do this because they treat both inflammation, tau, and amyloid, or maybe they can help the amyloid antibodies work better. So we will be dosing in this clinical trial once every 12 weeks. It's a single and multiple ascending dose clinical trial. We'll be doing the study in Israel, in the Netherlands, and in the United Kingdom. And we're very, very excited. The study is in its startup phases right now. And we're very, very excited and hoping to be able to deliver data, maybe even by next year's CTAD or meeting shortly thereafter. Obviously, want to thank the team at IBC, Professor Michal Schwartz, who was the originator of this technology. But this is just a really, really, really exciting time to be involved in Alzheimer's clinical development. And hopefully we at IBC, at Immunobrain Checkpoint, with IBC-Ab002 and others, can continue to bring more and better therapies to our patients with Alzheimer's disease and other neurodegenerative diseases as well. And I mention other neurodegenerative diseases because, as I pointed out, our mechanism also reduces tau in the brain. And so this mechanism may be applicable to tauopathies like frontotemporal dementias and progressive supranuclear palsy and other diseases that cause as much suffering as does Alzheimer's disease. So we're also looking forward to moving in that direction. And I know there are a number of experts at the meeting here and in the audience who are interested in these disorders.